Well, first of all, thank you very much for the invitation, Philippe. I'm very happy to present you this stuff today. This is one of the situations where actually the title of the conference really influenced me a lot in what I will present. It's the first time I'm going to present you this. I'm going to present this stuff, not to you, but all together. And what I will try to do today, in part because of the title of the conference, is try to discuss how we think about the relation between DC function in mice and humans. I was probably invited as a mouse man and not as a man of both. I will present you very little about mice and then discuss what we have been doing. Actually, when I say we, is one bright postdoc in the lab that joined the lab something like 2 years ago called Elodie Segura, who has been trying to understand how DC subsets specialize functionally in humans. But still, I will start with this general introduction to disease, which is, probably useless because of what just Paula told you and the beautiful introduction she gave to you. Our thinking about this is that dendritic cells are different. They are phagocytes, so they basically eat up particles in the body, but they are very different in function overall when you compare them to conventional phagocytes like macrophages and neutrophils. These guys are mainly involved in innate immunity. They destroy pathogens, they participate to inflammation. While the way we think about dendritic cells overall globally is that they are more involved into regulating immune responses in general and adaptive immune responses in particular. And to do this, they specialize in presenting antigens to T cells among other functions. Basically, dendritic cells are under a state in tissues and secondary lymph organs, and they respond to stress, to pathogens, to tissue damage, using, and you saw that in Paula's talk, a set of different developmental programs that bring them to be able to interact in secondary lymphoid organs with different effector cells of the innate and adaptive immune system. Initially, we all thought that this was a linear, so they existed in an immature, we used to call it a mature state, and that you needed this transformation for disease to become functional. Actually, now the picture is much more complex, and I borrowed this image from a Gaetano Reis de Sousa review a few years ago, just to illustrate that disease can respond in very different ways, and they can engage different developmental programs to become cells with different functions in terms of immunity, being able to start IL-17 type responses or Th2 or Th1 or different types of immune response. Now, this is already, the way they do this and the plasticity required to do this is not very well understood, but this level of complexity was, in the last few years, increased in a very big way by the fact that we now know that there are a lot of DC subsets. And usually, including me, DC subsets are the time during talks where you can go to sleep and then think about something else and relax. And that is probably because, in part, we didn't know what all these subsets were doing. They had a lot of markers and phenotypical characteristics. But going into it now, I managed not to sleep at least during my own talks on DC subsets. So this is how we think about subsets in mice. There are, first of all, conventional or myeloid dendritic cells and plasmacytoid dendritic cells. And then among the myeloid ones, there are several different types of cells. The ones that we call migratory, which reside in tissues and then migrate towards secondary lymphoid organs, including lymph nodes. There are Langerhans cells and dermal cells, and you will, in the skin, and you will hear a much more detailed description of all these subsets in a little while when Florent will present his talk. And then there are resident dendritic cells that live always in secondary lymphoid organs, and in mice, the two main subsets are distinguished by the expression of CD8 alpha. And then, so these are the skin subsets that then migrate here, and I will not go into great detail into that. And then in human, we really knew very little about these subsets. Most of the work had been done in the skin, where things are evolving rapidly, but most of the work had been done in blood. And in blood, you find myeloid disease and plasmacytoid disease, and among myeloid disease, you find two main subsets, which are distinguished by the expression of BDCA1 and BDCA3 together with CLEC9A. So these two subsets are present in blood, and they are thought to correspond to the resident that I just told you about in mice, the resident dendritic cells in spleen and tonsils. Okay, in humans, lymph nodes had never been analyzed, and a couple of years ago when we started all this, we thought we needed to know what disease in lymph nodes look like, and that is what we started doing a few years ago. And I will not present you all the work. It was published this year, but basically just the conclusions that we had from that work. Basically, what we found is that in lymph nodes, in human lymph nodes, and these are lymph nodes from cancer patients, the lymph nodes are not invaded, and the patients have not been treated, but they are still cancer patients, so that limits the conclusions that we can make. So we found resident dendritic cells and migratory dendritic cells. These were skin-draining lymph nodes, and we compared them to lymph nodes that did not drain the skin. Among resident dendritic cells, we found plasmacytoid dendritic cells, BDCA1 positive, and CLEC9 positive or BDCA3 positive cells, which correspond, all these resident ones, to the ones that are found in the blood. However, functionally, the cells are not similar. In the blood, in particular, these two BDCA1 and CLEC9 positive DCs are cycling, but they are not cycling in lymph nodes, and their functional capacities are also different. So they somehow, these in the blood, we think, represent precursors of these guys here that become fully functional and mature in lymph nodes. And then we found three main populations of migratory dendritic cells: the CD1A positive that come from skin, Langerhans cells that come from skin, and then a population of mannose receptor positive cells that also come from skin. Now, the one thing we did is try to isolate all these guys and then put them together with T cells to have some hint on their functions. And this is the kind of results that we obtained when we. So these are the ex vivo purified dendritic cell subsets compared to lymph nodes and blood, and then incubated together with naive CD4 positive T cells from the blood, and then analyze secretion of interferon gamma or IL-5. And what you can see is that cells in the blood are very good interferon gamma inducers, not IL-5. Actually, we also tested other Th2 cytokines, and these guys in the blood basically induce Th1-like responses from naive T lymphocytes. In contrary, these CD1A migratory DCs, they are very good at inducing IL-5 and very poor for interferon gamma, and so on. You see how the different, some are just the same for the two. We also, so in doing this in different kinds of settings and with different cytokines, the conclusions look like this. Basically, resident dendritic cells are induce both Th1 and Th2. They don't distinguish very well among the two. Sorry, I will have some water. They don't distinguish very well, and they are not very selective between Th1 and Th2, but they are poor inducer of CXCL13, which is a marker of T follicular helper cells in humans. In contrast, this guy here, which this mannose receptor positive disease that are migratory, they are very good inducers of T helper follicular cell-like phenotype, but not of Th1, Th2. The two other migratory cell types mainly induce Th2. I don't think this is, I don't know to what extent this is relevant functionally in a true immune response. It just says that at the steady state, in the absence of any other stimulation, we just put the cells with T cells, with naive T cells ex vivo, they do have different functional abilities. Now, another thing that was done in mouse and that we thought was interesting to do in humans is the analysis of a different situation when you have inflammation in the organism. And in mice, it was shown by different groups, including Ralph Steinman recently, that there is a population of monocyte-derived inflammatory dendritic cells that comes up, mainly in the spleen and lymph nodes, that have a particular phenotype, but their function was not very well characterized yet. In humans, we knew very little about this. There were some reports showing DC populations under inflammatory and infection conditions in the skin and in the circulation, but we knew nothing about any other localization. So what we did in humans is take two sources of inflammatory fluid from patients. The first one is synovial fluid from arthritis patients, and the second one is ovary ascites from ovarian cancer patients. Both are inflammatory sites, a lot of IL-6 and activated cells, including Th17 T cells. And we isolated myeloid cells defined a strategy to gate these cells and characterize two main populations of cells. One that expresses BDCA1 and it has a dendritic cell-like phenotype. We did a lot of other markers to correlate this, and this population seems to be very homogeneous when we analyze all other markers that we could look at. And then a population of CD16 positive cells, BDCA1 negative, that really look like macrophages and have high phagocytic capacity and other characteristics of macrophages. One way you can easily distinguish these two guys is using MLR. So if you incubate the cells ex vivo with naive CD4 positive T cells and then look for MLR for division, you see that the BDCA1 are much more efficient than the macrophage-like cells, which was actually the initial definition that Ralph Steinman proposed to distinguish dendritic cells and macrophages. So I will only show you some of the results that we got analyzing these. We did a transcriptome analysis comparing inflammatory dendritic cells and inflammatory macrophages from ascitic fluid and from synovial fluid, and we compared those to BDCA1 positive disease from blood and to the two main populations of monocytes in the blood in order to try to distinguish what we were looking at here. This is a PCA, a principal component analysis, just to show you that inflammatory dendritic cells and inflammatory macrophages behave, so each dot here is one population. They behave as distinct independent populations of cells. They are related to each other more than they are related to the other ones. And of course, it's very difficult to know if that is because of the inflammatory environment that is present here and not here, or because the two cells are related in terms of ontogeny. And actually, to address that question, we did another set of analysis together with Marc Dallot, and I maybe will not present you the details of this because I'm not sure I understand them very well. But we compared all these populations that I just showed you to published internet profiles, including from monocyte-derived dendritic cells. And the one population that resembled the most to inflammatory ex vivo purified dendritic cells is monocyte-derived dendritic cells. So those were the two best matches. These monocytes, so when you compare them to macrophages to other populations, they were less homolog. And so what we concluded from these is that actually the inflammatory dendritic cells are derived from macrophages. That is the most easy explanation from all our results. Like in mice, they differentiate from the blood into the inflammatory tissue into two distinct, so monocytes differentiate into two distinct populations, either macrophage-like or dendritic cell-like, and we don't really know yet what are the determinants for that. What we also did in this case is try to understand a little bit about their phenotype, their function, and the one thing that popped out, and again I'm only showing you a small part of the result, is that if you incubate these purified dendritic cell inflammatory or blood dendritic cells actually here, the one thing that comes up is that these inflammatory dendritic cells produce T cells that secrete high levels of IL-17, which is not the case with the others. Here we are comparing, for example, with interferon gamma, and the blood disease, again, are very good for interferon gamma, but not all these inflammatory cells. We also tried to understand why this was so, and we analyzed these guys now in the absence of any T cells, just activating them in vitro to see what they secrete that could account for this induction of IL-17. And again, what came up is that these inflammatory dendritic cells are the only ones that secrete IL-23 in vitro when you activate them here with PAM3 or CD40. Macrophages fail to have non-detected levels of IL-13. They both, of course, secrete other inflammatory cytokines, including TNF, IL-6, IL-1 here. So basically, what we conclude from this is that in humans, inflammatory fluids contain a distinct population of dendritic cells that are different from activated or inflammatory macrophages that induce IL-17 secretion from naive CD4s in an MLR reaction, and that secrete IL-23 after activation consistent with this phenotype here. We also think that inflammatory disease in humans, like in mice, derive from monocytes, and they are, because they are the best homologs of in vitro derived monocyte dendritic cells. So all this part, we kept the idea that actually when we went to humans, the findings in mice were good predictors, that actually mouse, as far as dendritic cells were concerned up to here, were quite similar in terms of organization of the DC part. And I will show you that that is incorrect for what I will show you now, which concerns cross-presentation. So let me first introduce MHC class 1 presentation. So endogenous presentation, as you know, concerns mainly misfolded or defective ribosome, misfolded proteins or defective ribosome products, which are degraded by the proteasome in the cytosol of the presenting cell. And this results in peptides that are translocated into the ER and loaded on MHC class 1 for transport to the surface. So this is the conventional endogenous MHC class 1 presentation pathway. Now, for quite some years now, we know that dendritic cells do something else. They also cross-present antigen. What this means is that they also present antigens on MHC class 1, which are derived from the extracellular milieu that are endocytosed or phagocytosed by cells. There is a big debate around this, not the existence of cross-presentation, which is now, I think, hard to question, but about the importance of this process during immune responses. And we still don't have a good way to evaluate that because we don't have any way to specifically inhibit cross-presentation in any model. Every time we do that, we also inhibit other processes. So we still don't really know how important this is physiologically, but the more and more experiments come up, the more the importance of this process seems to be real, but we still don't know when it's under question. Now, there are, if you think about this process of cross-presentation, at least two very important steps in terms of cell biology that allow cross-presentation to occur. The first one is that you need to limit degradation of exogenous antigens. When the cells degrade antigens too efficiently, they just destroy them. There is no cross-presentation. That is, for example, what happens in macrophages. The second important step is the transfer to the cytosol. Antigens need to get from endocytic compartments into the cytosol, and there is a lot of evidence for that. And we now designed a method to measure this step quite precisely. I will show you a little bit in a minute. But it is very important to do that efficiently if you want to cross-present antigens. And in mice, it was actually shown that there is a specialization for these functions among DC subsets. So among DC subsets, the ones that cross-present antigens efficiently are particularly CD8 positive resident dendritic cells in spleen. The CD103 migrating this from dendritic cells in skin and lung, and inflammatory dendritic cells. Also, these ones seem to do it in a different way in terms of cell biology. I will not go into that. But what was more studied in more detail by us and by others is that these guys here have specialized their endocytic pathway for these functions. They maintain an alkaline or neutral pH in their phagosomes, and thereby they decrease the rate of degradation and allow cross-presentation to be efficient. And they also have very efficient machinery that we still ignore the molecular nature of to transfer their antigens into the cytosol. So if you compare these two cells here, there is a very clear-cut difference in the way their endocytic and phagocytic pathways are organized. And this is, in this model, I think this is one of the main reasons why we think that these functions here are important for cross-presentation. We have ways to modify genetically these two functions, at least, sorry, this function here, the alkaline and the degradation rate in phagosomes, and then we can affect and show that the difference between these two guys is due to this kind of difference. Now, in humans, we knew very little about cross-presentation until recently in mouse subsets. In monocyte-derived dendritic cells, they cross-present like human cells do. But there were a group of papers 2 years ago showing first that among myeloid dendritic cells in the blood, the CLEG9A BDCA3 positive subset represents the equivalent or the homologue to murine CD8 positive disease, and that was based on transcriptome analysis and homology between the transcriptional programs of the two cells. And then we also know that the dermal CD1A positive dendritic cells cross-present antigens efficiently, and this is from the work of different groups, including Jacques Bonchereau a while ago. Now, we thought it was important, again, to go to dendritic cells in vivo and to study what was going on. The first thing we did was to generate CD1A positive and CD13 skin-like. So these are generated in vitro, just to validate this notion that low degradation and transport to the cytosol were important for cross-presentation. We knew that CD1A were the ones that cross-presented and not the CD14. So we generated the two cells, we sorted them, and then we analyzed cross-presentation in a particular system using a long peptide for melan A antigen. And what you see here is that indeed, as expected, the CD1A positive disease PDCs generated in vitro cross-present and not the CD14. So we use this system to validate our cell biology assays in humans, and this is just the summary. I will not show you these results. Like in mouse, CD1A cross-present soluble antigen, like mouse CD8 positives and CD14s don't. The pH in phagosomes, like in mice, is neutral in CD1A, like in CD8 positives, and acidic in CD14s. The production of ROS in phagosomes is the same. This is one of the ways dendritic cells prevent acidification of their phagosomes. And then the important one is here, transfer to the cytosol, and again, only the CD1A positive transport antigen to the cytosol and not the CD14 positive. So this just validated our assays and this notion that we defined in mice that cross-presenting cells in humans also have these distinct intrinsic characteristics in the phagocytic pathway. And then we went to the true subpopulations. We used human tonsil because that was the only source that we could get. So these are tonsils from healthy donors. We isolated 4 populations: PDCs, macrophages, BDCA1, and PLEG9, BDCA3 positive cells from the tonsil. There are no migratory disease in tonsil, as you know. These are the proportions of the 4 populations that we get. There are a lot of PDCs and few PLEG9A positive cells, over 18 donors. And this is the cross-presentation assay. Very surprisingly, because of those papers I just mentioned that proposed that the BDCA3 were specialized for cross-presentation, we didn't find any difference in the efficiency of cross-presentation among these 3 populations here. Macrophages failed to significantly cross-present antigen. Here you have the cross-presentation with the long peptide. Here you have the short peptide that doesn't need any processing, so it's a control that the cells can activate the T-cell clone. So no selectivity. When we did the same experiment, but this time using necrotic cells as a food for antigen cross-presentation, we still didn't see a big difference between these 2 guys here. The PDCs failed to phagocytose the necrotic cells, so they failed to cross-present like the macrophages. The macrophages did phagocytose a lot, but still did not cross-present. But we didn't see selectivity among these 2 guys here. So actually, it's not completely true. If I show you all the donors with the necrotic cells, you can see that there were 3 donors that were low in the BDCA1 cells for this cross-presentation of necrotic cells, and that it was induced, it was higher in the BDCA3, and 2 donors where there was no difference. So there seems to be heterogeneity, but certainly not a complete specialization of the cells for cross-presentation as we see in mice. We therefore decided to analyze their cross-presentation, the phagocytic pathway, using the same assays that we used before for CD1A and CD14, and for the mouse CD8 positive and CD8 negative. Actually, it was done by others in PDCs. But what we found is basically no difference between BDCA1, BDCA3, and PDCs in the cross-presentation ability for soluble antigen, at least, in the pH, in phagosomes for BDCA3 and BDCA1. They are both neutral. And in the transfer to the cytosol, no difference here using a quantitative assay that we developed. So basically, what this says is that also the BDCA3 PLEG9 positive disease seem to be the homologs in terms of transcriptome program. We don't see a different ability of dendritic cells to cross-present, and actually they all do it efficiently. So it looks like in humans, unlike mice, even PDCs are efficient to cross-present antigens. So the functional specialization for cross-presentation that we observe in mice does not seem to occur in humans. This is contrary to what has been published, and we can discuss that in a minute, but everything that has been published until now was done in the blood and not in lymphoid organs, and that, I think, is mainly one of the reasons why this happens. So here is the conclusion for this last part. All resident disease subtypes in tonsils, but non-macrophages, cross-present soluble antigens to CD8 positive T cells. All resident subtypes in tonsils, but not macrophages, display phagocytic functions compatible with efficient cross-presentation, showing that our predictions from mice were not all true. Thank you very much for your attention. I just want to point out the name of the person that did almost all the experiments that I show you, Elodie Segurin. A lot of this could be done thanks to the group of Vassili Soumelis that set up a lot of the circuits to get all the organs and the cells in Institut Curie, and we collaborated with different people for different aspects of the study. And I thank you very much for your attention.